The Coywolf. The Coywolf did not require human intervention to exist. Humans simply created the conditions, and the rest happened faster than anyone expected. For 3 centuries, European settlers hunted wolves across North America toward extinction. The surviving wolves pushed out of their territory, moved south and east into coyote range. Under normal conditions, wolves and coyotes rarely produce offspring together. The size difference makes it difficult. Their territories rarely overlap enough to make it likely, but with wolf populations collapsing, the conditions changed. The size gap between the 2 animals narrowed across generations of smaller, isolated wolves. Hybridization became viable. The resulting animal inherited the wolf's larger frame and cooperative hunting intelligence. It also inherited the coyote's behavioral flexibility, the ability to eat almost anything and live almost anywhere. The Coywolf runs at speeds competitive with a greyhound. It weighs roughly the same as a German Shepherd. Its bite force exceeds most domestic dog breeds. It's not a coyote that got slightly larger. It is a wolf-level predator that has learned to live in apartment density neighborhoods, and it now occupies every major city in North America, including New York, Boston, and Toronto. Then researchers ran the DNA. What they found was not a 2-way hybrid. Coy wolves in the northeastern United States carry a 3rd genetic signature, domestic dog. The animal moving through Central Park at night is part wolf, part coyote, and part dog. A 3-way hybrid that assembled itself across 3 centuries without any human planning or awareness. No one designed this animal, no one predicted it. It built itself from the wreckage of the wolf population that humans dismantled. The coy wolf is a hybrid that formed from the conditions humans produced. What comes next required something different. The slow disappearance of an entire ecosystem. The Grolar Bear. The Grolar Bear should not exist. Until 2006, most biologists agreed it probably didn't. Grizzly bears and polar bears have been separate species for roughly 500,000 years. Geography kept them apart for that entire span. Grizzlies in boreal forests, polar bears on Arctic sea ice. The separation was stable. It held through ice ages, through mass extinctions, through every major climate shift in the last 500,000 years. Then the sea ice began retreating faster than at any point in that history. Polar bears hunt ringed seals from sea ice platforms. As those platforms shrink and disappear earlier each year, polar bears move south in search of food, directly into grizzly territory. The 2 species are genetically close enough that their offspring are viable. More than viable. They are fertile. A Grolar Bear can reproduce. It is not a biological dead end. It is the beginning of a lineage. A Grolar Bear carries the polar bear's mass, one of the largest land predators on Earth, combined with the grizzly's ability to survive on a land-based omnivore diet. Berries, roots, ground squirrels, carrion. It is a bear built for an Arctic that no longer exists, equipped with the digestive system of the environment that replaced it. 2nd generation Grolar bears have been confirmed in the wild. The hybridization is not a series of isolated accidents between lost individuals. It is compounding. The Grolar Bear is in the process of becoming a self-sustaining population, a new bear assembled in real time from 2 existing ones in a habitat that neither parent species originally occupied. The Grolar Bear is a hybrid produced by a habitat disappearing faster than any biological response is possible. What comes next is a hybrid produced by something stranger, a conflict in the rules of reproductive biology itself. The Wholphin off the coast of Hawaii in a research facility tank. There is an animal that moves through the water like a dolphin, but is built like something else entirely. Its size is wrong, its coloring is wrong, its chromosome count is wrong, it should not be fertile. It is. The Wholphin exists at a genetic distance that reproductive biology says should make it impossible, and then goes further by producing offspring of its own. False killer whales whales and bottlenose dolphins are not close relatives. They belong to different genera, a taxonomic gap wider than the one separating lions and tigers. Their chromosome counts differ by one pair. In most species, a single chromosome mismatch is enough to produce offspring that are either non-viable or permanently sterile. The genetic machinery for cell division cannot reconcile the difference. In cetaceans, whales, and dolphins, something about the reproductive biology appears to bridge this gap. Researchers have not fully explained why. The result was Kekai Malu, born at Sea Life Park in Hawaii in 1985. She was intermediate in size between her parents, with coloring that matched neither. She was fertile. The chromosome gap between a Wholphin's parents is roughly equivalent to the genetic distance between a human and a chimpanzee. That gap in primates is an uncrossable reproductive barrier. In cetaceans, it produced an animal that went on to have calves of her own, a hybrid of two animals from different genera, a combination that reproductive biology had effectively ruled out, became a grandmother. The rule did not hold. The Wholphin broke a rule about chromosomes. The next animal broke something more fundamental. It broke the rule about what an animal is supposed to look like when its two parent species have never, across millions of years of parallel existence, produced the same jaw. The Narluga. In 1990, a hunter in Greenland shot what he thought was an unusually shaped beluga. The skull sat in a museum for nearly 30 years before anyone understood what it was. The Narluga is not a confirmed living species. It is a single skull held at the Natural History Museum of Denmark that took 3 decades and a DNA laboratory to identify. Narwhals and belugas are the only 2 members of their entire taxonomic family. They are the closest living relatives available to each other. They share Arctic range, they occasionally share feeding grounds, but physical hybridization between them is rare enough that the possibility would not considered when the skull arrived at the museum. Staff assumed it was a deformity, a malformed beluga, a misidentification. When Danish geneticists ran DNA analysis in 2019, the result was unambiguous. The animal was a 50-50 narwhal-beluga cross, confirmed by the genetic signatures of both species present in equal measure. The skull's dentition matched no known species. The narwhal has a single spiraling tusk. The beluga has multiple small teeth. The narluga had several small, slightly spiraled teeth arranged in a jaw that combined both gene sets, producing an anatomy that neither parent species has in any form across their entire biological history. Now, isotope analysis of the bone revealed something further. The narluga had been eating primarily bottom-dwelling fish. Narwhals do not typically eat bottom-dwelling fish. Belugas do not typically eat bottom-dwelling fish. The hybrid had developed feed-in behavior that belonged to neither parent, occupying an ecological role that did not exist before the animal did. It was not filling a gap, it was creating one. The Zebroid. The Zebroid looks like a horse. It moves like a horse. It is built like a horse. Then you notice the legs, striped unmistakably in the pattern of an animal that has never been domesticated in thousands of years of human attempts. Zebras and horses belong to the same biological family. Their chromosome counts differ. A horse carries 64, a plain zebra carries 44, but the gap is not wide enough to prevent reproduction. The Zebroid is the result, and it inherits something from each parent that cannot be separated out. From the horse, the body shape, the size, the general temperament of an animal that has lived alongside humans for millennia. From from the zebra, the stripes concentrated on the legs and neck, and something else entirely, like the behavioral wiring of a wild animal that has never been tamed. A zebroid cannot be treated like a horse. Handlers who approach it the way they would a domestic horse often find that the zebra instinct activates without warning the flight response, the kick, the bite. Veterinary procedures that are routine in horses require chemical sedation in zebroids. The body is domestic. The wiring is not. The two systems exist in the same animal simultaneously with no resolution between them. Male zebroids are sterile. The mismatched chromosome sets cannot pair correctly during the cell division required for reproduction. The animal is a biological one-way door. It can be produced, but it cannot continue itself. Every zebroid that exists had to be deliberately created. None of them will produce another. The kama. The camel and the llama have not shared a continent for roughly 3 million years. One spent that time in the deserts of the Middle East and North Africa. The other spent it in the high Andes of South America. They are distant relatives with no mechanism under any natural condition to meet. In 1998, at the Camel Reproduction Center in Dubai, a male dromedary camel and a female llama produced an offspring. Not through natural mating. A dromedary outweighs a llama by a factor of 6, making that physically impossible through artificial insemination. The resulting animal, named Rama, was the first confirmed cross between the Old World and New World branches of the camel family. Two lineages that had been separated since before the formation of the modern Sahara Desert. The Kama inherited the camel's short ears and long tail. It inherited the llama's cloven hooves. It had no hump. Its gestation had lasted somewhere between the llama's 11 months and the camel's 13. A biological compromise the two reproductive systems reached without any precedent to work from. When Rama reached maturity, he showed interest in llamas and guanaco and showed the camel's temperament rather than the llama's. The project had aimed to produce a large, strong, wool-producing animal with a cooperative disposition, and what it produced was smaller than expected and considerably less cooperative. The Kama represents something the other animals in this video do not. A hybrid that required human technology to exist. Geography had separated its parent species for millions of years. No natural event was going to close that distance. It took a laboratory in the Arabian desert to run an experiment that 3 million years of separation had made impossible by every other means. The hybrid shark. In 2012, a research team conducting a routine survey of shark populations along the eastern coast of Australia found 57 sharks that looked like one species but tested as another. Genetic analysis confirmed what the physical data had suggested and then complicated. The animals were not one species or the other. They were both. The Australian black-tip shark and the common black-tip shark are sister species, closely related, sharing overlapping ranges along Australia's coast, but distinct enough that interbreeding between them was considered essentially impossible. Sharks reproduce through internal fertilization with specific mating behaviors that were thought to act as a reliable barrier between species. The 57 specimens proved that barrier had broken down. Some were first-generation hybrids, others were second and third generation, the offspring of hybrids that had themselves bred successfully. The hybridization was not a single event. It was a population. What the hybrids had that neither parent species possessed alone was range. The Australian blacktip is restricted to warm tropical waters. The common blacktip operates in cooler, temperate zones. The hybrid could survive in both. Its territory stretched over 2,000 kilometers of Australian coastline, from tropical waters in the north to cooler southern waters that neither parent species could tolerate. tolerate alone. It was not simply a combination of two animals. It was an animal with a habitat that had not existed before it did. These were the first shark hybrids ever identified by science. How long they had been hybridizing before anyone noticed is unknown. The genetic tools to detect them are recent. The hybridization itself may not be. The liger. Everything covered so far involves animals that crossed a barrier. During geographic, chromosomal, taxonomic, the liger did not cross a barrier, and it fell into a gap that no prior biological combination had ever produced because no prior biological combination ever needed to close it. What happens inside a liger's cells is not hybridization in the conventional sense. It is a conflict between two genetic systems that has no resolution and no ceiling. Lions and tigers carry what geneticists call genomic imprinting, genes that are switched on or off based on which parent contributed them. In lions, the paternal growth genes are active. Cubs from different mothers compete for resources, and larger cubs survive more of those competitions. In tigers, the maternal growth limiting genes are active. A female raises cubs alone, and cubs that grow too large too quickly exhaust her resources. When a male lion breeds with a female tiger, the cub inherits the lion's active growth genes, but not the tiger's growth limiting ones. The break is disengaged. There is no mechanism to stop growth at the point where a normal lion stops, or where a normal tiger stops, or at any point in between. The animal continues past every size threshold the cat family has ever produced. The largest confirmed liger weighed roughly the same as a concert grand piano. It stood at the shoulder height of a draft horse. One exists in a zoo in the United States right now. The experiment that produced it was not designed. The follow-on experiment has simply never been run.